Okay, so the Unit 1 for biology, the first unit in biology, the first question on the test says, draw lines to identify the cell type to which each of these organelles belongs. So some organelles may be present in multiple cells. So there's 6 marks, and you've got 1, 2, 3, 4, 5, 6 there. So we're looking for these to join up with maybe more than one of these. So nucleoid is present in a prokaryote. We got the Golgi apparatus, both animal and plant, eukaryotic. The 70S, which is the smaller type of ribosome, only in prokaryotes. The centrioles, that's the one thing animals have that plants don't. Other than that, plants got everything else that plants have got, that animals have got, sorry. And then you got the 80S ribosomes, which is on the rough ER. Both animal and plants has those. And then the tonoplast, only present in the plant cell there. An electron micrograph of an animal cell is shown below. Now it tells us a little bit about this, because at first you look at this, you don't know what's going on. But the fact that this says nucleus, so the whole thing here is the nucleus, C must be the nucleolus. Let's just push that up there. So the first one I went for was C, which was the nucleolus, and then I can start working it out then. So coming off the nucleus is the ER, so B is either going to be the rough or the smooth. It's likely to be the smooth there, but because it's sort of closer into the nucleus, but they'll accept any one. And then this little structure here is a mitochondria. Okay, it's not going to be a chloroplast because it's an animal cell, so that's your 3 marks there. Okay, this one, don't get confused because they're not asking you to talk about gram staining here. This is just preparing a light microscope. So if you would have done examples with onion cells or even a cheek cell, that's all you've got to change here is the fact that it's going to be a blood cell. So it's 4 marks, and for each good point I would suggest maybe they're worth about a half a mark. So first thing I've said is produce a smear of blood on the slide. Then you put the cover slip above the slide. You place the slide on the stage. You select a low magnification lens. You always start with a low first. You illuminate the slide, so you've got to get some light underneath, and then adjust the 2 focuses. That's, you know, a bit of a mark there, and then adjust the magnification if necessary. If you were looking at an onion cell, you'd have to add a stain to be able to see this blood, but in this case we don't have to. And then the next question says, what is the function? Now, a lot of students answer this with, so they'll say that B is a chloroplast, A is a mitochondria, and C is the rough ER. But it says the function. What is the function of the organelles? So you don't actually give what they are. It's just the site of respiration for A, which is the mitochondria, produces energy for the cell. Then B is going to be the site of photosynthesis, and then C is going to be the site of protein synthesis, which is the rough ER. They'll also accept there if you put the site or transport and synthesis of lipids or carbohydrates, because it's not that clear whether it's the smooth ER or the rough ER. I mean, it looks to be the outer, towards the outer ER, so it's going to be the rough ER there, but they'll accept any answers for that. Now we come on to gram staining. It says bacteria cells can be classified as either gram-negative or gram-positive using a test. What part of the bacterial cell determines the result of the test? Well, it's the cell wall. Okay, so negative and positive have different compositions on the outer layers on the cell wall there. How is this test performed? So heat a sample to the slide, that fixes it. So you fix the sample to the slide, the bacteria that is. Then you apply a stain. So in this first case it's crystal violet. It's gonna turn it purple. Then apply iodine solution. That's going to form a complex with the crystal violet. And then you decolorize it using ethanol. So if it washes off, if it goes clear, it's likely to be gram stain negative, because once you add that counter stain as the final step, which is saffronin, and it should turn pink. if it's positive, it won't turn pink, and it's all to do with the cell wall composition. I think negative has two membranes. It's got a thin wall with two membranes surrounding it, and the positive has just got that thick wall and then the membrane on the inside. And then the last question there on this one: how can the result of the test be used to decide the method for treatment? Well, gram-stain positives are more responsive to antibiotics, the purple ones, whereas the gram-stain negatives are resistant to antibiotics, so they need another treatment. And that's about the detail. You need to go in for 2 marks there. And then the last question is magnification, and should be easy to pick up marks on magnification so long as you get plenty of practice on this. So, the first misconception is they're not giving you the size of that line there, okay? That is a scale saying that this line is worth 120 micrometers. So what you need to do, so that's the actual size, so that's your A. Your image size, you have to measure. So I've measured that to be 5 centimeters or 50 millimeters. It's up to you how you convert. If you're dealing in millimeters, then there's 1,000 micrometers for every 1 millimeter. If you're dealing with centimeters, then there's going to be 10,000 micrometers in every 1 centimeter, because there's 10 millimeters in a centimeter. So you need to add a 0 there. So the first thing we need to do is to convert that. Well, the first thing we need to do is to use our triangle. So if we're covering, so we're asked to find the magnification, you put your finger over M, so M equals I over A. So I've got that, I picked up my first mark there. So M equals I over A, which is 5 centimeters over 120 micrometers. Because the units are different, I need to get the units right. Best way to do that is to times the image size to get it into micrometers. So it's always going to be a bigger number. There's always like more micrometers in a centimeter. So there's always going to be a bigger number. So you multiply. So it's going to be 50,000 over 120, and that gives you your magnification there. This X should be in front of that. Okay, I didn't leave enough space there. That's really naughty. I wouldn't get the mark for that. So it's going to be X 416.6. And then question B there, the image below shows blood cells. It's Magnus was telling us the magnification this time. Look, well, they've done the same there. They've put the X after. I'm sure you have to put the X in front. I mean, maybe it'll work both ways. But definitely, the way I've learned it is put the X in front. So, again, we measure this. So now we know the magnification. We know the image size because we measure it to be 1 centimeter or 10 millimeters. That 10 millimeters becomes 10,000 micrometers by timesing it by 1,000 or 10,000, depending on which unit you're starting with. If it's a centimeter, I put my finger over because it wants to know calculate the true size, which is the actual size of the cell labeled X. So actual, cover your thumb or cover your finger over the A, is I over M. So A equals I over M. That's your 1 mark. By converting and giving you the right equation there, you get another mark, so 10,000 over 1,000. And then you get another mark there for the answer, 10 micrometers. And that is the answer to that one, 3 marks.